Hello everyone, can you, am I audible? Just message if I am, and then we can proceed ahead. Just give me a minute. Um, can you view my, the presentation, which is written Revision 1 MOLGEN, Fundamentals and Introduction to Sequence Analysis. Can you hear? Okay, good. So I will quickly go through the lecture, and what I'm trying to do in this one is that I will mostly focus on the fundamentals, so that it will be a review of whatever we have already learned, and then I would also like you to learn something that is about the sequence analysis, which will help you gain skills in the molecular genetics. So the first thing is, I hope you remember the chromatin organization. We have discussed it a little bit, and we are going to discuss it a little more on this side. We know the structure of DNA. It is double-stranded. It is the phosphate backbones are on the outside, and the bases are in the in a ladder, steps in a ladder. And then you know the Chargaff's rules: A base pairs with T and G base pairs with C. AT has 2 hydrogen bonds and GC has 3 hydrogen bonds. So that is a helix, and per turn there are about 10.5 base pairs, and there are different forms of DNA, A, B, Z, and they all have different dimensions. In majority of the cases, what we find is that the DNA is in B form. So it has somewhere around 10.5 base pairs per 1 turn. And the size of 1 turn is 34 angstroms, or you can say it's a 3.4 nanometers. So the DNA, as you can see in this case, the DNA here that is present as a, 1 second, I'll try to switch this on. Yes, so the DNA that is present in the cell will always be, in every cell, the genetic material is double-stranded DNA. Only in some plasmids you may have single-stranded DNA. In some in phages or viruses you may have some single-stranded DNA or double-stranded, or it could be in viruses and viroids. In some viruses and viroids you will have RNA as the genetic material. Okay, so if we see the organization of the genome in the how as a physical entity, how it is being packed is what we are trying to look at. So these are the details about the DNA. Chargaff's rules important, some measurements, the base pairing is important, and then I hope you can recollect who has discovered the transforming principle. Can you recollect? You can always keep messaging so that I can have a look at it. If you said Griffith, that is right. Griffith has discovered the transforming principle, but who was the first one to have identified that the transforming principle is DNA? And how did they, what is the name of that experiment they have done? Or you can say they have done, no, not Hershey, every McCarthy McLeod. Yes, that is right. They have used RNases, proteases, and DNases, and seen which one fails to transform a R strain, that is rough pneumonial strain, into a smooth one. And then we have the Hershey and Chase. The blender experiment proved that bacteriophages have DNA as the genetic material, and so on. So those experiments are also important. I think I have given you the video lectures before, so it should be available to you. And then the next part, we have learned about nucleosomes. In the nucleosomes, what are all the histones that we know of? There are 5 different histones. The first one is called as the there are 2 different classes or types. One are core histones and the other one is linker histone. What is the linker histone? Yes, that is right. I see that there are a lot of answers and they're all right. H1 is the linker histone. H2A, H2B, H3 and H4 are core histones. H3 and H4 form a tetramere, and H2A and H2B forms a dimer. So you have 2 dimers of H2A, H2B, H2A, H2B, and then 1 tetramer of H3, H4. And that means in total, in one nucleosome, that you have 8 histone molecules in total. Okay, so you have 2 H2A, 2 H2B, 2 H3, and 2 H4. And the if you have H1, it usually binds in between like this. It is not a part of the nucleosome. And those are some of the things. And you have furthermore compaction. That is right. You can say is a nucleosome as an octamer, heterooctamer made up of 4 different types of histones. So in the chromatin, so the once this nucleosomes, it is like nucleosome. Sorry, you have a bead of strings. That's what a string of with lot of beads. That's nucleosomes, and that can fold upon itself into a 30 nanometer chromatin. So you can form something like this. There are 2 different forms: solenoid and zigzag. Solenoid is much more. You can see the visuals in the textbook. It has much more compact. It is compact, but at the same time, it is amenable to opening up. Whereas zigzag crisscrosses and it is a little bit a tight organization. And this 30 nanometer chromatin fiber again loops on itself, and that further loops into a 700 nanometers condensed chromatin loop. And during metaphase, I hope you know what is metaphase. What is the most important thing? What are the most important characteristics that happen during metaphase? Yes, you have metaphase plate. That is, it is not. All the chromosomes are condensed. That is the first thing that you can see. And normally, you will not see a chromosome in this way. You will see this kind of chromosome only when it is when it has condensed completely. And there are 2 different proteins that are required for this condensation. Can anybody say which proteins are important for? Yes, condensin. Deepa said condensin is right. There is also one more. Yes, cohesin is also right. Cohesin is the one that puts the 2 daughter chromatids. So if you see that in the cell cycle, you have G1, and then you have S phase. During S phase, synthesis of or the replication happens, and then you have G2 phase, and then you have mitosis phase. During S phase, what was one chromosome becomes into 2 chromosomes, and these 2 are now called as sister chromatids. They're still bound together, and that protein that keeps these 2 together is cohesin. And the chromosome, if it is a long one and it is made into a shorter compressed one, that is done by condensin. I think the names themselves say that, and we don't have to remember that for too long time. Yes, I think most of you have got the answers, and that's right. Adhesin is not an answer, so you might have to correct cohesin and condensin. Correct me if I was wrong. I hope this is what you should try to understand, and I already told you before. If you take, we have 23 different chromosomes. We have 2 sets of them, so we have 46 chromosomes in each of our cells, except in red blood cells. And in megakaryoplasts, you have many more. We have majority of our cells are diploid, so we have 2 sets, one inherited from our father, one from the mother. So these we have 22 allo chromosomes, which are and then you have one sex chromosome, or if you're talking in diploid, two sex chromosomes, and you have 44 allo chromosomes or somatic chromosomes. If you are a girl, you will have two X chromosomes. If you are a boy, you will have one X and one Y chromosome. I hope you will try to remember those things, and those are fundamental, and is a good way to start these things. We will move on to the next one, and what we will discuss is something about what makes. We are now going into, yeah, before I go in, I think I was telling you this. We have so many chromosomes, and if we put all the chromosomes extended completely and we put it as linearly, one end attached to this, something like this, you will have approximately 2 meters of DNA. That is, 2 meters of DNA comes to this much, and you're compacting it into something like 15 nanometers or 15 micrometers of nucleus. Imagine 2 meters of DNA being compacted into something that is invisible. That is the engineering that evolution has come about. The next one is about the genetic information, how it is organized. In viruses, you can have, I hope you remember what is an ORF. You can type it quickly before I keep moving ahead. So yes, open reading frame, but then what defines an open reading frame? That is, it has to start with, it has a start codon, and then continuous uninterrupted codons, and ends with a stop codon. Can you recollect what is a start codon? Yes, if you can recollect, AUG, always use capital letters, don't use small letters. I understand some of you might be using, yes. And then AUG is the start codon, or if you're talking in terms of DNA, it is ATG. And what are the three stop codons? Quickly. Yes, UAA, UAG, and UGA. So those are the stop codons. From the start to the stop codon, the codons that are present in between, they should not have any gap. And codon is made up of 3 bases. Okay, we will see it in the next slides. But for now, what I want you to understand is that how much space is occupied for each gene. Viruses, as I always mention, they are some of the most efficient in the sense that even though they have a very short genome size, they pack a lot of genetic information into it. And that's why it is viruses are always important. So you can have, they have overlapping genes like this. That is one, this is one ORF which will produce a protein, and this is the second one which will produce the second protein. You may also have other ORFs reading on the downstream, which will produce a different protein. So that is how viruses, the gene density you call it, that is number of genes in a unit length of DNA. And the gene density is usually very high for the gene density is also very high for viruses. About diversity, we will speak of in viruses. We will speak of later. Diversity, they can be as huge. You can take any kind of living form, that is cellular living forms, bacteria, archaea, fungi, protists, plants, animals, anything. You have their respective viruses. That means their the genetic information of viruses is so advanced in a way that they can feed on the cells. In bacteria, the differences, we are trying to different the we are trying to different. I will get to the diversity of the proteins in a few minutes. So what I'm trying to do in this slide is compare between viruses, bacteria, and eukaryotes. So that will be a good way of trying to remember what we have learned so far. So in viruses, the genome size is small. They produce but they produce good enough number of proteins compared to their size, relative to the size. In bacteria, the architecture is little different. There might be overlapping little bit, but not as much as in viruses. So in bacteria, they are polycistronic. Can you recollect what is polycistron? What is a cistron, first of all? So you can take it as ORF as one equal to one gene, which can also be referred to as one cistron. Okay, writing is not great, but just adjust with that. Can you tell me what is the difference between ORF gene and a cistron? ORF produces, usually it is about a production of a protein. If you're talking in terms of the end product, what is encoded, then ORF gene and cistron all are meaning the same thing. Okay, if you're saying if you're talking about genes that produce RNA, they do not have ORF. Okay, something like rRNA, for example, they do not have open reading frame. They just produce the rRNA and that is used. Same as the case with tRNA and small RNAs and so on. So cistron and gene, they imply that the product is either a gene, either a protein or an RNA. But if you're saying ORF, you are implicitly telling that it will produce the end product is a protein, not just the RNA. So that is the only difference. Anyway, with bacteria, when you say polycistronic, poly is many, and cistrons, you have gene 1, gene 2, and gene 3, or ORF1, ORF2, and ORF3. And they're all under the control of one promoter, and there are no terminators here. Assume that there is a terminator here that is usually represented something like this. So if this promoter allows the transcription to happen, then you will produce a mRNA that is having something like this, and each of it you will have ORF1 and ORF2 and ORF3. And this will produce, upon translation, this will produce a protein. This will produce the second protein, and from the ORF3, the third protein is produced. So from one mRNA, you're having 3 different proteins produced. So why is it something like this in bacteria? Usually, all the genes under one particular promoter are useful in the same. So, for example, you have seen, if you can recollect from LAC-Z, we have lacoperon. Okay, we have discussed about LAC-Z, LAC-Y, and LAC-A. All these 3 proteins are important, or enzymes are important for metabolizing lactose or bringing in the lactose. So it will be wise or logical for the bacteria to have expressed that one mRNA and produce all the related proteins to metabolize or biosynthesis proteins, if it is the case of tripoperon, for example, or arabinosoperon. So the next level of advancement is in eukaryotes. So in eukaryotes, they are monocistronic. What does that mean? That means one promoter encodes or produces one mRNA, which usually produces only one protein. Okay, but they are distributed into, or the genes are interrupted. What is an intron? That is a part of a non-coding region within the coding region. It is something like exon is, this is the start codon, this is where the start is, and this would have the stop codon here. The last exon 5 would have the stop codon. Exon 1 starts with the start codon, and then we have several regions like these, and which are referred to as intervening sequences, and they are introns. We don't know if they are completely or always junk. Some people do think that they may produce some other small RNAs or other things which might be. There are actually examples where there is some genetic information in the intron. So we cannot necessarily say that it is a junk, but majority does seem like it is junk. So what is the next step upon transcription? The transcription will produce what is it called? If you can remember, it's called a pre-mRNA. The pre-mRNA is then in the pre-mRNA is spliced or pre-mRNA is processed. What is splicing? Can anybody recollect what is the literal meaning of splicing? Is it cutting off introns or joining of exons? It is joining of exons, that's right. So as I said in the class 2, I think, cutting off introns does not mean joining of exons. But joining of exons, if you said, that means you're removing the introns. The objective is to produce a protein at the end that is in bits here. Unless you bring all the exons together, you cannot produce a functional protein. So in that light, why do you think, what is the advantage of having this thing? It is actually having introns is a burden. You are spending a lot of energy to synthesize it, to cut it out, and so on. What is, I need to look at one particular, yes, diversity is right, but I would also want the topic that we, what do you call that? We used a phrase as a part of a topic in this particular lesson. I would like you to recollect that. I'm looking for answers. It is called alternative splicing, right? What is alternative splicing? If you can remember, it is exon 1, 2, 3, 4, 5. If they all come together, they will produce a one type of protein. If you have exon 1, 2, and 3, that is a different kind of protein. Okay, it could be shorter, but it is still a different protein. Exon 1, 2, and 4 and 5. I mean, 3 is skipped. Exon 3 is skipped, then it is a different type of protein. So you have many types of combinations that can happen. One thing, however, will be that in eukaryotes. So I will talk about it like this, that viruses can produce, one mRNA can produce more than one protein, true or false. Bacterial mRNA can produce more than one protein, true or false. Eukaryotic can produce more than one type of proteins, true or false. It is true for almost all the cases. In viruses, it is producing two different types of protein. It can produce two different types of proteins because the ORFs are usually overlapping. Okay. In viruses, I would want you to focus on this one again. This promoter will produce one mRNA, and this one will produce another mRNA. These two are not related. Okay. This one and this promoter, they're different ones. So anyway, in this promoter, if this mRNA is what you're taking, it can produce two proteins. This mRNA will produce three proteins, and the last one will produce, the last one is going to produce, the eukaryotes also is going to produce that. So this one is produced, in viruses, two proteins are produced because it has two overlapping open reading frames. In bacteria, it is producing more than one type of protein because it has multiple cistrons on the same mRNA. And then eukaryotes, it can produce more than one type of protein through alternate splicing. What is the difference between the proteins produced from one bacterial mRNA and the proteins produced from one eukaryotic mRNA? If you can recollect that, we should be able to, we're doing good enough job. In bacteria, all the proteins are completely unrelated to each other. This one has absolutely, there will not be, that may not be any different, any similarity between these three proteins, this one, and this one. Whereas if a protein is produced through alternate splicing, there will be a lot of similarities between, because the reading frames are same and reading ORF, the exons are same. Only the combinations of the exons are changing. So you'll have a lot of similarities between the different types of proteins produced by through alternate splicing. Okay, and now I think now we have to discuss about the central dogma. This will give us a good or it will review we will review some things in there. So in here, DNA, and here I'm trying to directly go with the eukaryotes because it has the most complex conditions. So through replication, you will produce more copies of the DNA, and upon transcription, for transcription, you need what are all the things. And while I say that, I would want you to tell me the description of the RNA polymerase. How would you describe RNA polymerase and the substrates? Everything is in the figure, right? Transcription requires RNA polymerase and RNTPs. Okay. In what is the difference between prokaryotic transcription and eukaryotic transcription? Prokaryotic transcription and the RNA polymerases in prokaryotes are is a single RNA polymerase. And it is code transcription and translation are coupled in prokaryotes. Why is that? It is because there is no nuclear membrane or nucleus, and there is one another one. Yes. The processing of mRNA is different. There is not much of processing in prokaryotic mRNA, but eukaryotic mRNA have to be processed because there are introns. You need a 5' cap and a poly A tail in eukaryotes. And if you have to describe DNA polymerase, how do you describe that? Dependency and polymerase. I hope you remember these things that we have discussed. Yes, for DNA polymerase, you would say DNA dependent, DNA polymerase. In all of molecular biology, what we are talking, it is discussing, we are actually talking about information technology, genetic information technology. So if information has to move from one place or location to a different one, or from one entity to another one, or one molecule to another one, there should be template dependency. And that's why all the enzymes, the first of the enzymes that might have come, which were template dependent. DNA polymerase, it is template dependent. RNA polymerase is template dependent, and ribosome is also template dependent. And that is, the dependency is what makes the information flow from one molecule to the other molecule. So it's always a good practice to learn to describe any enzyme in terms of what its activity is, that is polymerization, which language it speaks, and the other one is about what is it copying from or what it is dependent on for the information. DNA polymerases are DNA dependent DNA polymerases. RNA polymerases are DNA dependent RNA polymerases. Ribosomes are RNA dependent amino acid polymerases, you can say that. And there is also reverse transcriptase. Anybody, you can type what is the reverse transcriptase in terms of dependency, and where do you find them? Can you give one or two examples? Yes, reverse transcriptase is RNA dependent DNA polymerase. When people write RNA or DNA, always use caps. Don't use small. Even if it takes a second delay, that is all right, but always put it in caps. Never unfollow or always follow the standards. Okay, good. So where do you find reverse transcriptase? We have discussed, yes, you can find several reverse transcriptases in retroviruses where they have RNA genome. They need to make a DNA copy and then they integrate into the DNA of the host genome. So they need reverse transcriptase. There are also one more I'm looking, which is present in majority of the eukaryotic. Yes, you may have retrovirus-like transposons. That is right, and telomerase RNA. That's right. Telomerase is also a reverse transcriptase. Telomerase is different because it has telomerase has protein component and an RNA component. RNA is used as the template, and protein component makes the polymerization. So it is a RNA dependent DNA polymerase. Before I forgot to ask you this: what is the function of centromere? What is the function of centromere? Can anybody discuss that? And also the function of telomeres. We know the function of telomeres. Function of telomeres, quickly. It is to protect the ends of the chromosomes. Okay, the function of telomeres is to protect the ends of the chromosomes. What is the function of centromere? Centromere holds the two sister chromatids together. Yes, but that's not necessarily only one. What is something that forms here on the centromere? What is it called as? Can you recollect anything from cell biology or also in molecular biology? That is right, it is called as kinetochore. What is the kinetochore important for? Yes, chromosome segregation. The two chromosome chromatids, sister chromatids, are attached like this. And they are attached through cohesin. And after a while, there is centromere. Assume this is the centromere part, and they are still attached. So here is a kinetochore, and here will be a kinetochore. This one is bound to a spindle fiber, and this one will be bound. During anaphase, the cohesin is broken up, and then the chromosomes will move from towards the opposite. This is pulled towards the other side. So centromere is important for the organization of kinetochores, and the kinetochores are important for the binding of the spindle fibers arising from the poles to the kinetochore and being pulled. That is chromosome segregation. Centromere is essential for chromosome segregation into the daughter cells, and that's why I hope you know what is aneuploidy. If not, please note it down. Aneuploidy. And tomorrow I would want that you all be prepared or you submit an assignment, aneuploidy and some of the diseases associated with aneuploidy in humans. Okay, let's get back to the central dogma in molecular biology. And splicing happens, and there are a lot of splicing proteins or spliceosomes, if they are called. You have a multi-protein and RNA molecules. They are large complexes. They recognize the 5' splice site and 3' splice site, and then they fold it upon and then they break it, and eventually binding exon 1 to exon 2. I have a question. This is RNA 1 and it has one exon, and this is another RNA. This one has exon 2, and this is say exon 3, and here is exon 1. If splicing happens between these two to join and make this, what do you call that phenomena as? If something is splicing has been made between two exons present on two different RNA molecules, what do you call that? That's right, it's called Listinger. That's right, it's called as trans-splicing. Okay. What is a lariat? Anybody? What is a lariat? This is a lariat. It is one of the products that is formed in which type of splicing ones? And maybe it is a good point that you know what is the junction present here, and in which cases would you find this one a spliced, I mean the end product of a intron after splicing. Yes, I think majority of your answers are right. I hope you have done it well. So try to recollect these things. And then we have translation. It is what type of ribosomes do you find in prokaryotes? It is, what are the units called as? If you can recollect, the ribosomes, they are made, there are two subunits. One is smaller and one is a larger one. I'm waiting for answers. Yes, 50S and 30S in prokaryotes. In bacteria, you have 50S and 30S. And what is S stand for? S is a something like a sedimentation coefficient in ultracentrifugation. It is Swedberg unit. That's right. So in prokaryotes, you have 30 and 50S subunits. Both together will form 70S subunit. Whereas in eukaryotes, you have 40 and 50S. Both together will form 80S type. If you look at the ribosomes of cell organelles, that is, cell organelles are in something like mitochondria and chloroplast, you look, they have much more similarity to that of prokaryotic ribosomes, which means that, which also strengthens the idea of the endosymbiotic theory. So there are, translation is a complex one and a very interesting one. I think the video lecture is complete. You have to please watch those for additional details. You should know about the ribosomes, the tRNA that is present, and tRNA is also a fantastic molecule which will make everything. What are the 4 or 5 arms that are present? This one is called as D loop. What does D stand for? And this is something like this. What does D stand for in tRNA? This is the anticodon arm. This is how the anticodon is usually present. D loop is for dihydrouridine. That is typically this. It is named after the dihydrouridine. That is, those are the modified bases that are present in the tRNA. And here you have pseudouridine. This is the variable loop, and then this is the anticodon and loop. This is the anticodon loop, and here you have the acceptor arm. So tRNA molecules are also fantastic because they are central to translate the language, the information from the language of nucleic acids to that of the language of proteins. And how it has evolved and how everything has evolved around it is still a fantastic area of research. So here you have a structure. What is this whole structure called as? Anybody can you type it in? I want you to also tell me some things that I have left out here. What would this end be? And what would this end be? And what would this end be? And what is this? What are the ends that are this one here? I will call it as P, Q, R, and S. Can you tell me what is P? Anybody? SP is 5 prime G cap. That's fantastic, Deepa. And then R, no. Yes, I want, yes. So P is 5 prime and Q is 3 prime of the mRNA. How can you say it is 5 prime and 3 prime? You have to assume that the ribosomes are moving from this in this direction. All reading of information, whether be it on DNA or RNA, it is always done 5' to 3'. All of polymerization also happens from 5' to 3'. So the ribosome, if it is moving on the mRNA, it is moving from the 5' side of the mRNA to the 3' side. How do you know that it is moving in this direction and not in this direction? How did you decide that? That is based on the length of the protein that has been produced here. If it is moving from here till here, it would have covered only say 3 or 4 codons. And that means the length of the protein would have been only 3 amino acids or so. You cannot have a long one. If you see the length of the protein here, it is increasing. That means these ribosomes must have been reading more number of codons as they are moving forward. And that is how you determine that the ribosome is moving in this direction. This whole structure is called as a polysome. Okay, the polysome is mRNA completely coated with a lot of ribosomes on it. That is called as a polysome. For translation details about it, I think the video lectures were elaborate. I would like you to have a look at it and be inspired for to be amazed at how complex and how elegantly things are done at the molecular level. So what is being described in this figure? Quickly, I would want this is depicting semi-conservative replication. I want to know which scientist figured it out and what was the technique used in that. Yes, semi-conservative. Matthew, I don't know. Yes, Messelson and Stahl, that's right. And what is the principle that they have used? They have used 2 different nitrogens, N14 and N15. N14 and N15, that's right. So N14 is lighter than N15. So they have given, I would not discuss this. This is also there in some of the videos that are there. So, but you have to understand the principle that they have used density gradient ultracentrifugation to distinguish what are the other models that are present for explanation that were proposed to explain the DNA replication. Can you recollect any? This one is the one that you are seeing on the screen is semi-conservative replication. The other one would have been conservative replication. And then there is also a third model that is called as that is called as disruptive or dispersive. Okay. So out of all these things through this experiment, what was proved that semi-conservative replication is correct. And so this is accepted and the others are rejected. So here is a DNA sequence shown in A, and you have to be very careful about finding out open reading frames. Open reading frame always starts with start codon, that is ATG, and here you have a stop codon, that is this. Sometimes you may also have overlapping ORFs, that is it starts from here, the reading frame is here, and then it goes continuous, and then you may have a stop. When you are asked for sequence analysis, you have to look very carefully about all the open reading frames that are present. There are also, it might also be in this direction. There are 2 strands here, strand 5' to 3', and the bottom is written 5' to 3' like this. So you have to look on the reverse or the complementary strand as well for any ORFs. So pay attention and try to do that. I think we have done enough exercises on this. We also had something on for CAA1. on this one. I would like to quickly go through some of the some mutations, and then I will just stop in a while. So here is a open reading frame of that encodes some 10 amino acids and a stop codon that is present here, start codon. And what is a nonsense mutation? That is, if any of the sense codons, say here it is coding for serine, TCG, but because there was a mutation here, which has now changed into TAG, it is become a stop codon. So in the what you should what kind of mutation is this? You can describe it, right? It is a point mutation because there is only one base change. It is a substitution. Point mutation can be insertion, deletion, or substitution, and here it is a substitution. What kind of substitution is it? Transition or transversion? It is transversion. It is transversion because a purine is replacing or substituting a pyrimidine. Okay, A and G are purines, T and C are pyrimidines. So A is replacing or substituting C, so it is a transversion. That is at the level of DNA sequence. When you talk in the level of protein sequence, it is a nonsense mutation because a sense codon has become a nonsense codon or a stop codon, because of which now the protein is truncated and it is very small, and often it is usually non-functional. Then you have a frame shift mutation, that is because of insertion of a one base, and it could be deletion as well. And the reading frame from here onwards, these two are same as the original X1, and from then on they have changed, and because of the shift in the frame, and there is now a new start codon. You see that here is TAG, which was in this T was in part of this codon here, and it encodes tyrosine, and here it is encoding the AGC is encoding serine. Because of one insertion in this case, now TAG is being read as one codon, because of which it is now a stop codon and there is truncation. What is important here is the definition. What all things cause frame shift mutation? Insertions or deletions that are not multiples of 3 will cause a shift in the frame, reading frame, and that is frame shift mutation. What you should understand is the whole, from here onwards, you see that the whole of the protein is different from the original one here. Until here, you see that they are almost same. The X1, it is same, and in this one, in X3 also it is same, until the frame shift happens. From there onwards, they are entirely different proteins. And then there are also silent mutations, because although there is a change in the nucleotide sequence, there is no change in the protein sequence. Why is that? Why does that happen? That happens because both ACG and AGT, they both code for serine. Yes, that is right. The genetic code is degenerative. That means not all the cases will be, as we'll see in the next slide. Some bases, some amino acids are coded by more than one codon, and because of which silent mutations can happen. I want to know, what is an allele? What is an allele? Have you come across alleles? In Mendel's, we must have learned. Yes, that is right. So you can have a gene called, in this case, as we have seen, as I'm showing you, X1. I'll say X as one gene, and that gene can have different forms. When I say different forms, it means they have differences in their nucleotide sequence. Often, because of that nucleotide sequence, there will be changes in the nucleotide sequences. They may have phenotypic differences as well. So alleles are different forms of the same genes. It's worth remembering it that way. So here for X, I have discussed this X1 as the normal one, assume like that. And here is an X2, X3 is another allele, X4 is another allele. In these X2 and X3, usually it might be a loss of function, whereas X4 is again, there is no alteration in the phenotype, although there is a difference in the genotype and phenotype. Yes, in the genetic code, some of the amino acids are hyper, there is a lot of degeneracy in the sense serine, for example, is encoded by 6 codons. Here is 4 and here is 3. And arginine is coded by 6 codons. It is also having 6. Glycine has 4. And are there any amino acids that are just coded by 1 amino acid? None. Almost all the wait, well, let's see, trip, is there anywhere tryptophan? Trip is having 1 here. Okay, methionine, there will be another one also, I think. Where is methionine? Okay, methionine, do you see any other methionine anywhere else? No. Trip and methionine are having one codon each. So the genetic code is almost universal, but not absolute in different forms. Say in mitochondria, the genetic code is a little bit altered. There are differences between archaeal genetic code and bacterial genetic code. There are differences between prokaryotic and eukaryotic also. What you should understand is there are a lot of similarities, because of which we can say that this whole translation thing is evidence for evolution. And then there is diversification. There are modifications to the genetic code. And that's why you see differences in different organisms. So I don't need you to mug up this, but as I always maintain, it's important that you know that the start codon, AUG, and these 3 stop codons, UAA, UAG, and UGA. So this is the last slide. I'll stop with this. So in what is essential, or how you, if you have gained real knowledge from this course, you should be able to do some skill. You should have gained some skills with that. Your skills are about, I don't want you to learn things as a mugging up. We should not mug up. We should have a little higher level of education, in the sense that the skill required is about your calculations. So in the future, maybe in the end sem also, I might also ask you, I will give you a complex picture sequence, and then I will ask you to do all these things that are here. The first one is about dimensions. You know about the dimensions, length in millimeters, centimeters. I might ask anything. You know what is the size of space occupied by one, length occupied by one base pair in lengthwise. Can you quickly say that? Yes, 3.4 angstroms is right. 34 angstroms is wrong. 10, yes, that's right. 3.4 is correct. So all those calculations you should have done. Molecular weight you should have known, and similarly you should also have done the hydrogen bonds between the 2 strands. For this, if you have to do, you should have known the GC content and topology, linking number, and so on. We are not discussing so much of topology and Reith and other things, but at least linking number you should know. Nucleosomes and number of each of these histones, whether it is, if the question is about tetramers or dimers, you should be able to answer in that. And number of phosphate bonds broken for polymerization. Can you think of it? So if you have 10 base 10 say next ATP should be added, A should be added. So there are 3 phosphates. How many phosphate bonds are broken? So this is say adenine here, ATP or DATP if I'm talking about DNA. So this would be alpha, this is beta, and this is gamma. So the first thing is there should be a hydrogen bond breaking, the phosphate bond should be broken here. Yes, one phosphate bond is right, but you should also remember that we also break this pyrophosphate also. What is the enzyme involved in breaking the pyrophosphate? Simple thing, the enzyme name is called as pyrophosphatase. The energy released during the breakage of these beta and gamma phosphates is also used to drive the polymerization reaction. So the number of bonds broken for every addition is 2 phosphate bonds are broken. Remember that. You have to count everything that will allow the polymerization to happen. So if I said, okay, I'll give you a quick question and try to do that. 10 ATPs or DATPs are polymerized. How many phosphate bonds are broken? Quickly. I'll repeat the question. There are 10 DATPs and they're all polymerized now. Okay, into one single strand. How many are how many phosphate bonds are broken? Think critically. Think critically. How many phosphate bonds are made? If I ask you, what would you say? You have 1, 2, 3, 4, 5, 6, 7, 8, 9. Right? That's right. 9 is correct. This the one on the 5 prime end will have its phosphates like that intact. They are not broken. This is what is this is 1, 2, 3, 4, 5, 6, 7, 8, 9 will be made. 9 bonds are made. That means you should not count into this. So it would have been 9 into 2, that will be 18 phosphate bonds. Okay? Don't leave anything untouched. Think very critically, think very deep. Okay, that's very important all the time. Yes. Next, in the sequences, so the question usually comes with 2 different sequences, sequence 1 and sequence 2. You are making a comparison between these 2 sequences in all of these ways. Okay, you should look at how these 2 sequences are differing in terms of dimensions, bonds, and so on. We did this exercise for CAA 1, and it is going to come for NSEM also, I think. And then you also have translation and transcription level. I mean, you have to find out the number of ORFs present. There are also certain sequences I've put in the websites. I will send this slide to you. You all have to keep it with you and try to squeeze as much information possible from the sequence. The sequence that you're seeing on the screen now is very small one. The next one, the ones that I'll give you for exams will be larger. So you can read through these, and if you have any questions, you can come about. I will send this PPT now. Now, any questions or what should we do tomorrow? I would dedicate one class for any important topics that do not have video lectures. You can make those, but I cannot cover all in 1 hour. So I will choose. You can email me if you have on the Google Classroom. Please email me if you have any questions or you want some topics to be discussed. I'll try to work on that. Yes, before I leave, this will determine your lot of your skill. Sequence architecture and mutations. When you are comparing, you should describe what kind of mutation it is, the consequence of those mutations. You should also predict how that mutation may have happened, what might have caused that mutation. And you should also tell how that mutation may be repaired if it has happened, and if it is single standard assume how it can be repaired. Okay, so I will send this as such. You try to do all your analysis. It's a good practice. If you don't do it, you will each of you will get a different sequence. There is nothing like the whole class will not get one sequence. That's not going to happen. So, and you also have to, you should be able to discuss the, I mean, design the primers. We have discussed that in the class as well as in the lab, I think. If not, you watch some videos and we will see if we have to discuss that. And this is one important thing I want you to also do it. There is, I will send you some videos, find out how to use that. And then maybe tomorrow I'll try to introduce you this part. Remind me if I forgot to do that. Okay, any questions? Hungry? I hope you are hungry. Yes. Okay then. I'll see you tomorrow. Okay, ask question. Yes, Ritik, please ask. This will be the last question for today. Okay. Uh, See you. Have fun, and we'll stop this now. Okay. Uh, Bye, everybody. Have fun.